Mitosis is defined as the process of nuclear division that maintains chromosome number. This cell division takes place and produces two daughter cells that have the same type of number and chromosomes that the parent cells have. The chromosomes of each daughter cell have the same characteristics, such as same length and genes. This is called homologous chromosome.
Interphase is the resting phase between successive mitotic divisions of a cell, or between the first and second division of meiosis. The homologous chromosomes are distributed through two phases, G1 and G2. G1 is when the chromosomes are duplicated, and G2 is when each one has two double-stranded DNA molecules. These are called the sister chromatids. S phase is the phase of the cell cycle in which DNA is replicated. It occurs between G1 and G2.
Meiosis is the nuclear division that halves the chromosome number in the reproductive cells. Meiosis is sexual reproduction of organisms, reducing the number of chromosomes and gametes. Mitosis and meiosis are similar because they both include cell division. Although mitosis produces two identical daughter cells, meiosis is different, as it produces cells that only contain half as much DNA and are unique between themselves. However, during mitosis, the sister chromatids are pulled apart and end up in the same separate nuclei.
The four stages of mitosis are prophase, metaphase, anaphase, and telophase. Prophase is the process in which the genetic fibers in the cell's nucleus, called the chromatin, start to pack together and condense. Metaphase begins when the microtubules in the kinetochore attach to the sister chromatid centromere during prometaphase. Then the microtubules begin to pull on the sister chromatids, and eventually they shift to the center of the cell. Anaphase takes place when the centromeres are split up into the sister chromatids. Then the daughter cells begin, and the microtubules that make up the spindle begin to shrink. Lastly, telophase occurs when the new daughter cells get their own identical sets of chromosomes. Then a nuclear envelope forms around the daughter cell's new nuclei.
The first stage of meiosis is metaphase 1. This is when the homologous chromosome pairs align midway between the spindle poles. Then there's anaphase 1. This is when the homologous chromosomes separate and begin heading toward the spindle poles. The next telophase 1 is when a nuclear envelope forms around each cluster, so two haploid nuclei form. Next there is prophase 2. This is when meiosis is the most similar to mitosis. Duplicated chromosomes are separated out. Telophase 2, the cells pinch in the center and then divide again. Crossing over occurs during prophase 1. This is important because it allows for homologous chromosomes to separate, and the segregation of the chromosomes occurs during metaphase 1.
Cytokinesis is a cytoplasmic division that happens between late anaphase and the end of telophase. However, cytokinesis is not part of mitosis. Cytokinesis in animal cells is when a cleavage furrow forms and then proceeds to pinch the cell into two. In plant cells, cytokinesis is when a new cell wall inserts itself and separates the daughter cell's nuclei. Mitosis